Hello, I'm back, and it's time for human biology again. Today's lecture is number 19, and I'm titling it Biotechnology. So we're still talking about genetics, but now we're going to discuss how the science of genetics can be applied to modify organisms or the products they make. So this isn't just about understanding, it is about using it as a manipulative tool, and that's what biotechnology is. I've got the concepts of biology sections here, and I've got kind of a funny, maybe a scary, I'm not really sure, but there's a bunch of science going on here, and this science is somehow being used to produce, consider, manipulate some kind of organism. It looks like a sheep that's saying meow like a cat, or maybe it's just a cat that looks like a sheep. Don't really know, but clearly there's a little bit of interest and excitement and concern from the scientists over here, and there should be all of those things because there's a lot of that going on in this field of biotechnology. So let's see what we got. Okay, so what is biotechnology? Well, I got a bunch of definitions here. One from your text, OpenStack's Concepts of Biology, one from nature.com, and one from the UN, which is not a really scientific group, but it is a group that involves how science gets used around the world. So I think these are some pretty important definitions. It starts with bio, and so we expect it to be about organisms or cells or living systems. And here we have organisms or cells. Here we have organisms, cells, or cellular components. And here we have biological systems, living organisms, or derivatives thereof. All about biology. So we expect that. The other side of it is the technology side. So it's going to be using artificial methods to modify the genetic material of these things, of these organisms or cells, or developing new technologies, okay, or technological applications, modifying products or processes for specific use. We are turning life into tools. Tools to make better food for people, tools to make better medicine for people, tools to make people healthier, tools to sell products to people. All of this is considered biotechnology. If it involves a specific use and a manipulation of an organism or a cell, it's considered biotechnology. So it's a big category to talk about, and people look at it in all kinds of different ways. A couple of important points about it down below. It acknowledges the link between molecular and organismal scales. Obviously, the central dogma of molecular genetics says we get traits by having DNA, and it goes through these steps and gets made into traits. So if we can manipulate the DNA, we can get different traits. That is a foundational concept of biotechnology. It's going to involve manipulation. It has a history in selective breeding, which we have been doing for centuries, where farmers go out and they have some sheep or they have some corn, and they say, you know, that one sheep or that one stalk of corn really is better than all the rest. So let's get rid of all the rest and let's just grow that type. Let's just reproduce that type. That's selective breeding. And when we did that, we were selecting the DNA that we were going to get the traits from. So that was a form of biotechnology. And so that's where the history comes from. It's just that we've gotten way, way, way better at it by using all kinds of other techniques. And the primary goals are in medicine and agriculture. So let's move on from this definition. All right, well, here's some pictures that are about biotechnology, and some of these look a little scarier than just the simple definitions that we were talking about a minute ago. We've got a little preemie baby here being kept alive, likely from some biotechnological techniques. Not necessarily a bad thing, but some people might find that disturbing. We've got food that's being made that has better nutrition in it. Sometimes that's seen as a good thing. Sometimes it's seen as cultural manipulation. We have food with a barcode on it. That's not a real thing to have the barcode grown on, but it indicates that sometimes biotechnology is about profit and sometimes it's about ownership. Who owns the types of food? Who owns the types of medicine? And that's a big concern that people have. And when people have concerns, they get out in the streets and make a big stink about it. And these are people who don't like the idea of genetic manipulation. Money-hungry fascism are dead. I can't read it. Anyway, these are apparently people pretending to be the zombie apocalypse that comes along with biotechnology. It's controversial. None of these are truly wonderful. None of them are truly horrible. We will be sorting out the goods and bads of biotechnology for centuries to come. Okay, so we know that it's a fairly simple definition, but it can be applied by humans in lots of different ways. Sometimes we love it, sometimes we don't. But I want to talk to you about the techniques to get our understanding going here. And there's a lot of them. I just came up with some quick verbs that make it possible for you to see the diversity of different things that we might do with DNA to manipulate it and to make something that didn't exist before we manipulated it. We can isolate genes. We can purify them. We can amplify them, which is making extra copies of them. We can cut them. We can sort them. We can sequence them to get them to find out what the exact series of nucleotides is in those genes. We can splice them together. We can insert them where they don't belong. We can get them to be expressed when the normal body of an organism wouldn't be expressing them. We can compare them. We can test them. We can activate them. We can make them produce new traits. We can label them, track them. We can record them. We can make DNA sequences that never existed simply by putting nucleotides together. There is a lot of power in these verbs. These are not a simple list of the types of techniques, but this is a vision of how much power there is in the manipulation of DNA. And that ... is to indicate that this list could be a lot longer. And all we would have to do is dig around for some ideas here. And so I stuck a DNA molecule into the toolbox to start to think about this. Okay, so this slide is the essence of my lecture today. These are all different types of biotechnology techniques. And I'm going to introduce them, and I'm going to give an example of how they're done and try to wow you with what they are capable of. And by the end, I hope you say, wow, we can do that. And yes, the answer is yes, we can do that. So I am going to go through these here. Let's see, all the way to here. And then I'm going to do this last one. I'm going to leave out this, this group in the middle because they are going to be in the next lecture. They're all about computer analysis of genes, genomics, proteomics, bioinformatics, and metagenomics. Those I'm going to do later, but everything else with the purple around it, I'm going to talk about that today. And I'll come back to this slide because I think it's a good summary and I'll be able to look back at it when I get done with these. So let's get going. Okay, but before we get started, before we can do anything with DNA, we need to have some DNA. We need to acquire some DNA. So there's an organism somewhere. It's a person, it's a corn plant, it's a bacterium, some organism that has cells because cells, life, they go together. We're going to get some cells. We need to get the DNA out of those cells so that we can manipulate it. So here we have cells. Don't know what it is. It looks a little pink. Maybe it's some meat or something like that. We're going to take some cells and we're going to lyse them. L-Y-S-E. That just means bust them open. Sometimes we do it by grinding them. Sometimes we do it by adding solvents that break the outside of them up. But we're going to bust them up, okay? Then we're going to treat them with something that destroys proteins. DNA is not protein, right? So it's not going to be destroyed by whatever this is. But DNA is all held together in the nucleus by proteins. So we need to get rid of the proteins so that the DNA can be separated from all that. And that's what we're looking at here. We're using something called protease, which is an enzyme that breaks a protein. Nothing too fancy. And then our gloppy mess is going to separate into layers, and we're going to remove a layer that we know doesn't have any DNA in it. And then we're going to use something, it's usually very cold ethanol, to make the DNA solidify, to make it separate from the rest of the liquid. And when we do that, we can actually pull it out as a solid, and it's kind of sticky and gloppy. And so with this test tube, we're using a little glass rod, twisting it around in there, and the DNA sticks to it. Bam, we have a pure sample of DNA that came from that organism that the cells were from. And this is where we always start when we are trying to do a genetic test, when we are trying to figure out who did the crime, when we're trying to figure out who's the daddy, when we're trying to figure out how we can make better crops. All of these start by extracting DNA. Okay, so the first biotechnology technique I want to talk about is called polymerase chain reaction, PCR. Almost everything we're going to do starts with PCR, and that's because when we separate the DNA from the rest of the cell, it's usually a very, 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 very tiny amount. So we need to amplify it. We need to make copies of it. We make copies of it so that there's enough to actually do an analysis. So PCR is a process of making copies of a DNA sequence. And it's really pretty simple, so simple that now it used to be complicated lab technique, and now we have a machine that can do it for us. Like a copy machine, except that it's copying DNA. Not really like, like a paper copy machine, but this is how it works. DNA is in two strands that are held together, in a double helix, held together by those hydrogen bonds in the middle, and those are the weakest bonds. So if we have a strand, and it's all twisted around, and we heat it up a little bit, what happens is those bonds break first. So the two strands separate. Two single strands of DNA just because we heated them up. Now, they are drifting around in fluid now. They're apart from each other. So if we cool it down again, and there are some nucleotides, free nucleotides that are present in the fluid, these strands are not going to somehow find each other and bond back together again. No, the new nucleotides are going to jump into place and cover each of the single strands with a new synthesized strand, just like we learned in DNA synthesis. When we copied DNA in the nucleus, we're doing that in a machine instead. So we heat it up, we make sure there's some nucleotides in there, free nucleotides, and then we cool it down. And so then we've gone from one copy to two copies. If we heat it up again, each of those separate and form two new strands. So then if we've heated it up twice, we've got 4 copies. If we do it 3 times, we've got 8 copies. If we do it 4 times, we've got 16 copies. It is an exponential process, and so the number of copies that we make can be predicted by 2 to the power of the number of times we cycle through it. In fact, this process is called thermal cycling. Warm it up, cool it down. Warm it up, cool it down. Put some more nucleotides in. Warm it up, cool it down. Warm it up, cool it down. And as a result, we start with one double-stranded piece of DNA, and we're ending up with copies of that. And if we're going to do any kind of biotechnology technique, this is where we start, okay? We have the equipment to do this right here at Columbia College, so it's really not that complicated. PCR, polymerase chain reaction. All right, the next technique in our biotechnology toolbox is gel electrophoresis. Electro means there's electricity involved. Phoresis means movement of particles, and gel is where that movement is taking place. So here's why we do it. Here's what we're doing. It turns out that a DNA molecule has a slight negative charge, and negative things are attracted to positive things. So if we can create a current in a fluid, like in water or something like that, then the DNA will move toward the positive pole of that current. This is from some basic electrical stuff you learned probably in middle school, maybe in physics in high school. But we're just going to create a charge, and the DNA, because they're slightly negatively charged, will move toward the positive pole. But we don't want it to happen fast, and we want it to happen in a controlled way that allows us to sort out the pieces of DNA. So what we do is we send an electrical charge through a gel, not through water, not through some liquid that's moving around, but through a gel that allows things to move through it, but not really very fast. We make this gel out of seaweed. It's called agarose, and agar is a sugar that we get from seaweed. It's kind of like Jello, except Jello is made from the bones of cows and the skin of cows. So I'm thinking agarose is probably a better way to go. And so this gel is a fluid that allows DNA to very slowly pass through it if we send a current through this gel. But there are big pieces of DNA and there are small pieces of DNA. And the big pieces move more slowly because they have to work their way through the gel. They're not going to move through the small spaces as quickly as the small pieces. So what happens is, if we cut DNA up into a bunch of chunks, and then we put it in the gel and we send it through the gel, it will sort all those chunks according to size, because the small ones will move fast and the big ones will move slowly. They won't move as far. So we get these patterns of piles of DNA, and in this case, we put the DNA here at, whoops, wait a minute, I don't have a pen that's working. Okay, we put the DNA up here at this end, okay, in little holes in the gel. These little things here are little holes in the gel. And then we send the current through it, and so the DNA moves in this direction. The farther it moves, the smaller the pieces are. So the big pieces are way up in here, and the small pieces are way down in here. And what we can do is we can send materials that we know the sizes of the pieces. And for instance, there's this bright bar right here. We know exactly how big the molecules need to be to end up right there. And so we can say whether or not our samples actually have pieces of that size. So gel electrophoresis is sorting out chunks of DNA by size using a current that we run through a gel. Gel electrophoresis. The reason we can see these lights here that it looks bright is that we're putting dyes on them so that they will glow in the dark a little bit if we shine some fluorescent light on them so that we can actually see them. Or sometimes we put radioactive materials on the DNA and then put them on a piece of photographic paper because radiation will expose the photographic paper, and that might be what we're looking at here. But there are different ways to make it visible to us. DNA is not going to be visible to us in here. We either need to put a dye on it or a glowing fluorescent material in it or a radioactive material in it so that we can see these patterns, okay? So each one of these columns is actually a separate sample of DNA. And so from this, I would be able to conclude that in this sample right here, there are two different sizes of chunks of DNA. There's one that's a little bit bigger than the other because I see two bands, two big bands in that sample. So that's how we interpret this stuff. Okay, now we can extract some DNA from living cells. We can make copies of that DNA. We can chop up all those copies and then sort out all the fragments by size. That's with polymerase chain reaction, we make the copies, and with gel electrophoresis, we sort out the chunks by size. But how did we cut that DNA up? Did we get a tiny little knife and bring in the Ginsu chef? No. We don't use objects to cut up DNA. We use chemicals to cut up DNA. And the chemicals we use are made for us by bacteria. Bacteria make special enzymes that cut up DNA. We just have to get those enzymes out of the bacteria and we can use them as tools. These enzymes are called restriction enzymes. And they are really important for cutting DNA for a couple of reasons. First of all, they cut it in a very, very predictable manner because they only cut it in certain regions that are called palindromic sites. Palindromic. Have you heard what a palindrome is? It is a word that is read the same forwards as backwards, like mom is a palindrome, well, dad is also a palindrome. I'm sure there are some others that I could think of, maybe like pop or something like that. These are all words that are the same forward as backwards. A palindromic sequence of DNA reads the same on one strand in one direction as it does on the other strand in the other direction. So here's an example. This strand here, if we're moving with the arrow, says GGATCC. And if we look at the other strand, looking in the direction of the arrow, it says GGATCC. These restriction enzymes that bacteria make will recognize that, and they will cut it in that region. But they don't just cut it like right across. This is the beauty of restriction enzymes. They cut it between two letters on each of those strands such that they leave hanging chads on the ends. So we're going to cut it right here between these two G's, and then over here, right here between these two G's, allowing those strands to separate, and it creates what are called sticky ends. So not only have we cut this DNA in a very predictable manner, but we have created an end on this DNA, which will allow us to bring a new piece of DNA in that has that sequence on it and stick it to that end. This is how we splice pieces of DNA together. This is how we put DNA where it doesn't belong. We cut it using a restriction enzyme, and we splice together using DNA polymerase and using a sequence of DNA that we know has the right palindromic code on the end of it. Super cool. So polymerase chain reaction makes the copies, restriction enzymes cuts them up, gel electrophoresis sorts them out by size. That's where we are right now. Okay, so the next thing on my list of biotechnology techniques is called RIFLIP analysis. RFLP, pronounced RIFLIP. RIFLIP, say it with me, okay? What is RIFLIP? It is restriction fragment length polymorphism. Here we go, restriction fragment length polymorphism. Well, the word restriction is in there, so it involves restriction enzymes. Fragment length is in there, so we might use gel electrophoresis to sort out things by fragments. And polymorphism means basically all the different organisms are slightly different. If I take two organisms and extract some of their DNA and apply the same restriction enzymes to their DNA and sort out those pieces by using gel electrophoresis, they're going to produce slightly different patterns of fragment sizes. And so I can try to find out whether one sample of DNA is from the same organism or from a different organism, and I can try to find out how separately related they are. That's what RIFLIP analysis is, comparing two samples to see if they're from the same organism, the same individual, or how different they are. And that's what we're looking at here. We have a scale like a ruler, which we call the DNA ladder on the left. And then we have sample 1, sample 2, sample 3, sample 4. I don't know what these are from. They may be from separate organisms. Maybe they're from separate blood spots in a crime scene. Maybe they're from separate blood samples from people in the room. I don't know. But what it looks like to me is that these two samples right here are the same. They're showing the same banding pattern. And these two samples are the same. They're showing the same banding pattern. And this one is different from each of the other two. Just by doing that, we can make some conclusions about where these samples came from. RIFLIP analysis. We do the PCR, polymerase chain reaction. We apply the restriction enzymes. We sort them out by size, and we do it in a way that allows us to compare them. That's called RIFLIP analysis. Very cool. And if you do RIFLIP analysis on people or organisms to try to answer a question about how related they are, you are doing genetic forensics. Genetic forensics is RIFLIP analysis, specifically here on people, and we also call this DNA fingerprinting. Why DNA fingerprinting? Because it's about DNA, and because everybody's fingerprint is different, and everybody's banding pattern is different. Everybody's banding pattern. If we use the same restriction enzymes and the same gel electrophoresis, everybody's got a different banding pattern. The FBI uses 20 different loci where they use restriction enzymes to cut chunks of DNA out from 20 different places in the genome. They put it all together into a cocktail. They make copies of it. They break it up with restriction enzymes and sort it out, and everybody's got their own banding pattern. And what we're looking at here, just like in the last slide, is that numbers 6 and 7 are from the same individual. We know that because the chances of those banding patterns lining up that perfectly are minuscule compared to how many different arrangements of DNA that there might be. So I don't know if that means that that they've got maybe number 6 came from a blood sample and number 7 came from a crime scene. Maybe number 6 came from a baby and number 7 came from a paternity case. But we're trying to figure out if the DNA came from the same place. Okay, genetic forensics is RIFLIP analysis that's used to sort out individuals. Okay, that was very CSI, very Law and Order when we're looking at genetic forensics. I probably ought to mention that if you like those shows and you love it when the detectives look at the sample and say, send this off to the lab and get the DNA back as quick as you possibly can, and then it comes back in 10 minutes, that doesn't happen. It sometimes takes weeks to get these results because it has to be done carefully. It has to be done one step at a time. There's a backlog of other samples that are being done all the time. It just never happens the way it does on television. I just don't want to leave you with that impression. Maybe some of you are criminal justice students, and you probably ought to know that if you're going to be potentially asking for these kinds of services. Anyway, that's the genetic forensics. This is my next technique, and it's a kind of in a different area. It's called molecular cloning. Cloning sounds like copying, so it might be a little bit like polymerase chain reaction, but in this case, it's copying inside of a real organism. It's not a really exciting organism, it's bacteria. So what we're going to do is we're going to take DNA and be able to insert it into a bacterium, right? Because bacteria are capable of doing this on their own. We're just kind of manipulating their own behaviors. We insert it into a bacterium, and then we allow that bacterium to reproduce. And whatever the DNA makes in the bacterium, if it makes a protein that's really important to us, we can harvest that protein. All we have to do is grow the bacteria in a culture with some food, and we can gather the protein from those bacteria. So what we have here is we're going to culture some recombinant bacteria. Recombinant means we've inserted DNA from some other source. How do we do that? Well, bacteria make their own restriction enzymes. All we got to do is put the right ends on them, the sticky ends, and they will stick to the bacterial DNA, and then the bacteria will just grow and express their own genes. And the DNA where it's located is called a plasmid. Anyway, there's lots of things going on, but the point is that we can make stuff that's useful to us that we can grow just by growing the bacteria. Here's an example down here. I think this is insulin, okay? I don't know if you might have some experience with diabetes, but historically, to treat diabetes, we harvested insulin from pigs, from the pancreas of pigs, which was kind of a side product of the pork industry, the pig industry, and that was okay. But pig insulin and human insulin are not quite the same. So what we did was we worked out a methodology whereby we could take the gene for something like human insulin and insert it into bacteria. And then the bacteria will grow and produce that protein, that material. So now when diabetics get insulin, they get it from molecular cloning. And this picture here just shows that we're taking foreign DNA in a blob and inserting it into bacterial plasmids, and then we're going to grow the bacteria and make lots of colonies that have the trait we're interested in, and then we can just harvest that protein. It's pretty cool, molecular cloning. Okay, here is another useful technique. It is called marker probe hybridization, and I've already talked about it a little bit when I talked about the RIFLIP analysis and we put some radioactive material on those segments of DNA. That's basically a marker to show us where the DNA is and where it's not, so that we can visualize those bands. So RIFLIP analysis uses marker probe hybridization. Sometimes when we do the molecular cloning, we also have to mark certain bacteria that have the DNA that we're interested in, because we don't want to grow bacteria that don't contain the genes we're interested in. So in this case, we've got DNA growing in colonies in a dish, and the blue colonies started from bacterial cells that have the gene that we're interested in. So we can ignore the little white colonies and only harvest the little blue colonies. That is marking the DNA with a probe that gives it some color. And then over here, we have actually a picture of some cells, and we put a fluorescent probe on the DNA so that we can see little bits of DNA inside these cells that are actually fluorescing. We can tell which cells actually have the right DNA in them. So this happens with lots of different techniques. Whenever we have to sort target DNA out from DNA that is just kind of in the way, we're trying to find the target, we will put a color marker or a radioactive marker or a fluorescent marker on it so that we can sort it out easily. So that's called probe hybridization. Okay, so here is the next technique, and this is called sequencing. In this case, it's called chain termination sequencing. Sequencing is not about comparing two different pieces of DNA or solving a crime. What it is it's about taking a chunk of DNA and figuring out what the order of the nucleotides is in it. And we do that in order to look for certain genes. And we have this whole field called bioinformatics and genomics where we need to know those sequences. So when we don't know them from an organism or from a particular person, we need to have a way of sorting out that sequence of nucleotides. Remember, there are only 4 different nucleotides in DNA. So is it AGT, CCC, GTA, or is it GCTA, TCCA, GC? Anyway, there are all these different possible combinations. So it's really just a combination of some of the other techniques we've been doing. We use PCR, we use gel electrophoresis, but we break up the strands that we have into all of the possible lengths, every possible length. And so that includes, if I have a piece of DNA that's 10 nucleotides long, I'm going to have some pieces that are 9 nucleotides, some that are 8, some that are 7, some that are 6. We have all possible lengths, and we put markers on the last nucleotide. So we put them into gel electrophoresis, and we put a current through it, and we simply wait for the DNA to come out the other end of the gel. The first ones to come out are going to be the shortest ones. 1 nucleotide long, guanine. 2 nucleotides long, adenine. 3 nucleotides long, thymine. So now we have GAT. We are creating a sequence just based on how fast they end up reaching the end of the gel. By doing that, we can figure out what the sequence of nucleotides is. And it is very important in something called gene mapping. Gene mapping is how we take these chromosome pictures and say, look, this is where the gene for Fanconi anemia B is on chromosome X on the short arm, that's the P arm, at band number 22.2. That's how we map chromosomes. That's how we figure out what the locus of a gene is. Very cool. Chain termination sequencing. This is a major future science that could influence all of our medicine, all of our food. Okay, this is the last biotechnology technique I'm going to be discussing today. It is another form of cloning. You remember before we talked about molecular cloning, and that involved putting sequences of DNA into bacteria, allowing them to reproduce, and so they're making copies of the DNA for us. This is not that. This is not a molecular technique at all. It is actually the cloning of whole organisms. And it doesn't really fit in with a lot of the other techniques that I talked about because it doesn't involve PCR, it doesn't involve RIFLIP analysis or anything that's breaking up the chromosomes. What it involves is really, really, really tiny needles. It wasn't possible to do this technology until it was possible for us to make very, very small needles. The reason for that is that these needles have to be small enough to insert them into a cell to insert a new nucleus or insert some DNA into the nucleus. So we have a picture here of a cell, and it's being held in place by a little suction. That's what this is here. And we are inserting a needle and putting some DNA into that cell. Okay? Until we could make that kind of needle, this wasn't possible. Why do we have to do something like this? So that we can take a cell from an organism and take the DNA out of it, take the nucleus out of it, insert it into a different cell so that we can make a copy of the first organism by inserting the DNA into another one. This is reproductive cloning, not molecular cloning. It is insertion of a diploid nucleus into an egg that doesn't have a nucleus in it. You probably saw Jurassic Park. They put a little dinosaur DNA into, I think it was like a frog egg or something like that. And then here we have a sheep. You may have heard of Dolly. Dolly was a sheep that has been cloned. And here she is down here. And basically all we did was we took a nucleus out of a cell and inserted it into another cell so that we could then grow a copy of the original nucleus. And that's what Dolly was. This is, like I said, making a copy of an organism. We have done this with anything that's got a big egg is really easy to do. We've done it with lots of frogs. We've done it with fish. We've done it with mice. Clearly, we've done it with sheep. I have said humans with a question mark. I don't know how you feel about this, whether it's ethical or not. But if we can do it with a sheep, and you think that somebody's not doing this somewhere in the world right now with people, you are living in la la land, okay? I'm not saying it's a good idea. I'm just saying I would be shocked if it wasn't happening, because not everybody abides by the same scientific rules as the UN and the national scientist groups and people of that nature. Sometimes money can buy just about anything. So reproductive cloning. And I don't know, maybe you got a cat or a dog or a relative that you might be excited about doing this with. Clearly this could change the way we think about humanity in the future. And keep that in mind. There's Dolly. Okay, so here we are back at our review slide. As I said I would, I've been through a bunch of these. I will be talking about genomics and bioinformatics next time. But what I have talked about is polymerase chain reaction, which is how we make copies of DNA, gel electrophoresis, how we sort out fragments by size, restriction enzymes, which is how we break up those fragments, RIFLIP analysis, which is how we compare two samples based on restriction enzymes and gel electrophoresis, genetic forensics, which is how we solve the crime on Law and Order or on CSI, molecular cloning, which is how we make some medicines and different chemicals that are important by using bacteria to make them for us, marker and probe hybridization, how we isolate the DNA that we're interested in from the rest of the DNA that's not important, and how we determine a sequence of DNA by using gel electrophoresis to detect the nucleotides that are coming off of the end of a gel. And then the last thing was reproductive cloning. So I talked about every single one of those. I just want to say a couple things now about not just what is being done, but about why it's being done and what are the purposes of some of these major techniques. So often our focus gets on the whole forensics thing and using DNA to solve the crime. Clearly that's important. But this list has 3 other really important target goals that biotechnology has. And I just want to talk about each one of them and then maybe show some details after that. So the first one is genetic engineering. This is making new organisms that didn't exist before. More than just selective breeding, making organisms that have genes in them that make them grow faster, so that they produce more food, that make them healthier so that they don't die when there's some type of a disease outbreak. So we have, let's see, better agricultural efficiency, better agricultural nutrition. We can make fuels with biotechnology that turns crops into things like ethanol and fuels that can be used for different purposes. We can make medicines out of organisms, like for instance the molecular cloning technique, and we can do that with some other plants and animals that can produce these medicines for us. These are all examples of genetic engineering, putting genes into a living organism and having it make a product that we find useful. The next thing is called personalized medicine, and when you go to the doctor hears about your symptoms and says, this is what I think is going on, they give you a medicine. But this medicine might be given to anybody who has those symptoms. It turns out we're all different genetically, and personalized medicine means the doctor in the future may have to sort out some of your DNA before making a decision about what the best treatment is. Diagnosis and treatment based on genomic analysis. This is becoming more and more common. Pharmacogenetics is choosing a medicine based on not just your symptoms, but also your genes. And then obviously this is going to involve computers, and keeping track of all your genetics on computers will make it possible to line up different treatments with different genes. And so this is where we're going with biotechnology. The last one is called gene therapy. It would be great if somebody had a genetic abnormality like the ones we were talking about last time, and we could insert that gene into their body so then their cells would start making the protein that they weren't capable of making. Gene therapy is the way that we're hoping to get to that. And the major idea is that we could somehow insert a gene into a virus, which is not a living thing. It's a particle that takes over cells and forces those cells to make more viruses. But in this case, we would be loading the virus with DNA that makes something useful. Not more viruses, but something useful. So the idea would be that basically we can infect a person with particles that would allow their cells to now make new gene products. Kind of science fiction, but kind of exciting too. And this is where a lot of the people are looking for that cure for cancer, because cancer, remember, is a problem with the cell cycle. And if we could fix that by infecting a person, wow, that would be pretty cool. So let's remember these three things. Let me give you just some examples of them. Okay, so here's some examples of genetic engineering. These are transgenic organisms, which means organisms that contain genes that weren't there until we put them in. Here are some mice that have glow-in-the-dark jellyfish genes in them. Why? Because we can. I don't know whether they're just doing it to show that it's possible, but when we shine fluorescent lights on these mice, their ears and their eyes and their tails, any place where there's not a lot of hair, have this fluorescent jellyfish protein in them, and we use that as a biotechnology tool in a lot of places. So these are transgenic organisms. This is a rice which has some pigments in it that give it better nutritional value. It actually has more vitamin A in it, and so it's kind of yellow. And what we're trying to do is get Asian countries to grow this rice instead of the white rice that they're used to, because it would solve some problems with malnutrition. But it is, in fact, a transgenic organism. Down here we have an aquaculture salmon that grows faster than the wild salmon that the original genes were inserted into. It grows faster, so then it would make more muscle tissue quicker, and we would be able to eat them earlier. And then down here we have a fruit, which instead of growing lots of little seeds in it, grows one big one in the center, so that it's much easier to process when it comes time to eat it. These are all considered transgenic organisms, and quite frankly, most of the food that we go to the grocery store to buy is already transgenic in some ways. All of the produce has been selectively bred and is not really the wild type of produce. All of the meat has been not only bred, but has had genes inserted into the genetic line. So I know that some people are really nervous about GMO, genetically modified organisms, but I'm going to be honest with you, we have crossed that bridge, okay? It is rare to be eating food these days that you could not argue was already a genetically modified organism. And there's not a heck of a lot of evidence to show that it would have health influences on people that are consuming them. I would be much more nervous about things like pesticides that are used in association with them, or steroids, or maybe growth hormones or something like that. Those aren't necessarily about the genetic modification. It's about the proteins that they're making. Those things make me nervous. So those types of transgenic organisms, we definitely want to keep an eye on. But just because it grows faster doesn't mean it's dangerous. Genetic engineering. Okay, this is just to indicate that in addition to messing around with our food, we can use genetic engineering to grow corn that is used to make fuel, to grow algae that is used to make fuel, to grow bacteria that is used to make medicines. These are also applications of genetic engineering. Okay, the second thing on my list of goals for biotechnology was personalized healthcare. And today, all these people go to the doctor and they get a prescription. And sometimes that prescription works, sometimes it doesn't work, and sometimes it has terrible effects. There's diversity in that. In the future, what we hope is that these people go to the doctor, and because the doctor has all this information about DNA, from blood samples, about analysis that's on the computer, each of these groups of people get a slightly different prescription, a slightly different therapy, and it works better for everybody. That's the hope anyway. That's what personalized healthcare is. Okay, and last slide, and this is the last of the 3 goals of biotechnology that I introduced. This one is called gene therapy. And the idea here is that using a virus, this is not a coronavirus, it is an adenovirus. It has no envelope on the outside. Using a virus, we could load it with DNA and then introduce it into a person. And as a result, the virus would insert that DNA into the cell and would allow the cells in the body to make a new protein product from the new gene without actually having to take that protein in a medicine. That would be basically introducing the DNA to the patient. That's called gene therapy. It is definitely being worked on. There are not a lot of great success stories, but they're being very careful with it, as you might imagine, because it involves a virus, and we're a little bit sensitive about that right now. Anyway, that's gene therapy. I'm done. A little bit long today. But it is so long that I'm saying so long. I'll see you in class. Be safe.